Hello, I'm Jean-Gael speaking. I am a historian of neuroscience in Paris. I will be discussing with you today the history of the cells of the brain, including the first microscopic observations in the 17th century to present-day knowledge, including glial cell biology. First, Antony Philipp von Leeuwenhoek. What did he see in the brain? Globules? Fibers? As you know, he was a clothes merchant, and for his business he developed magnifying glasses to inspect his products. He made numerous observations of living material, and he became famous worldwide. He was finally invited to become a member of the Royal Society and of the French Académie des Sciences in Paris. He used to send the reports of his observation, to be translated into English at the Royal Society. On the left figure you can see the cross-section of a dried nerve from an oxen, where he observed cavities due to the process of desiccation, which he interpreted as canals where animal spirits would travel, according to the mechanical model imagined by Descartes. In the nerve cross-section of the middle image and the right image, Leeuwenhoek observed fat globules due to myelin decomposition. The middle image shows a section of a motor nerve from a sheep surrounded by fat. 5 nerve fibers are also visible as hollow cavities. The right image shows a section of a dried piece of pork brain. 2 blood vessels are visible. At high magnification, only fatty droplets are visible. The observations of Leeuwenhoek were met with great skepticism, since it was difficult to relate them to any hypothesis or theory of brain functions during his time. From an epistemological point of view, the drawings of Leeuwenhoek are realistic and not guided by theory, and therefore not theory-laden. His observations were motivated by curiosity and a religious faith and admiration for the minute creations of God. In the 18th century, Felice Fontana, an abbot, a naturalist and a physician, was also director of the Museum of Natural History of Florence, Italy. He dissociated nervous tissue in water in order to see its fine parts. He was able to make observations of fine fibers from nerves, which he published in 1781 in his treatise on viper venom. He observed fine fibers emerging from small nerves. He also observed globules, and his observations were also met with great skepticism, although the fine fibers he saw were not artifacts. In the 1830s, Christian Ehrenberg was professor of medicine at the Faculty of Medicine of Berlin. The famous Spanish histologist Ramón y Cajal considered he ought to be considered as the discoverer of nerve fibers. This is because he used good achromatic microscopes. He delacerated tissues and used microdissections from living invertebrates, thus being able to see living neurons. On the left image, he indeed reproduced his observations of globules from a ganglia of a leech, slightly crushed to increase its transparency. Ehrenberg observed globules which were nerve cells and not fatty artifacts. In the middle images, globules were found in the brain matter from cervical ganglia of insect Geotrupus. Bright spots are refringent nucleoles. In the right image up, nerve fibers from human nerve ganglia show globules, but fat droplets appear here, since the tissue used was not fresh. The right bottom image shows nerve fibers of the spinal cord of a red fish. Ehrenberg's observation demonstrated that all animals have the same kind of nervous globules, but such globules were not yet considered as cells. In the 1830s, Jan Evangelista Purkinje made important observations of nerve globules, called nerve cells, only a few years later. He was professor at the University of Breslau, now in Poland, where he founded a school of microscopy and where he directed a physiological institute. He published his grand memoir in 1857-1858. He observed globules of different kinds in the brain, piriform bodies in the cerebellum, rhomboid forms in the hippocampus, that is, pyramidal cells and tubes. He used acetic acid as fixator, methanol, oils, and tar for his preparations. The upper left image shows ganglionic bodies of the human thalamus, and upper right from human substantia nigra. The bottom left image shows pigmented and triangular ganglionic bodies of human oliver bodies. On the bottom right image, you can see ganglionic bodies of the cerebellum, still known today as Purkinje cells. However, the function of these globules were unclear. Robert Remarque was a Jewish-Polish-German pupil of Johannes Müller, the German head of the German Physiological School, and also a pupil of Ehrenberg in Berlin. In 1859, he became associate professor at the university in Berlin his life long. He probably never became full professor because of anti-Semitism, as was the case for other Jewish scientists from this history of brain cells. However, he was the first Jew to teach in the university in Berlin, at least in biology. But he was never fully recognized for his discoveries. In this work he made between 1836 and 1838, he observed nude fibers he called primitive fibers in all animals, see in the left image. Cellular endings are also seen in the image, and they are for remark of unknown origin. The left image shows globules with a nucleus and a nucleolus, thick endings corresponding to dendrites, and fibers with an envelope corresponding to myelinated fibers corresponding and showing a double membrane. The left image shows globules with a nucleus and nucleolus, thick endings corresponding to dendrites, and fibers with an envelope corresponding to myelinated fibers showing a double membrane. Thus, he described many of the parts of the nerve cells before the cell concept emerged. His drawings seem realistic and still not much influenced by any particular theory of brain function. He is now credited for this discovery of the physical link between neuronal somata, that is, Remarque's globules with nucleus and nucleolus, and on the one hand, protoplasmic processes, and on the other hand, primitive fibers, that is, both dendrites and axon. In 1837, the professor of botany at the University of Vienna, Matthias Leiden, observed that the nuclei of new plant cells originated from the nucleus of a mother cell. Discussing with his friend Schwann remembered he had previously made a similar observation in notochords, that is, the dorsal cartilaginous structure of chordate embryos. Both scientists acknowledged this parallel between plant cells and animal cells as a general fact, and Schwann established the cell theory accordingly, with its first law, according to which all living organisms are made up of cells and products arising from cells. Cell theory had a great impact in all domains of microscopic studies, including the histology of the brain. Adolf Hanover was a Danish anatomist and pathologist. He was a pupil of Johannes Müller and collaborator of Remarque. In 1840, he used chromatic acid to study the origins of nerve fibers. Inspired by cell theory and the work of Remarque, he was convinced that nerve fibers originated from nerve cells. In the left image, you can see large cells with nuclei, nucleoli, and endings from cells of the fourth ventricle of the human bulb, and also in a fish ganglion in the middle image. He could describe nerve fibers emerging from nerve cells in the right image, and a fiber with marrow leaking in A, fiber with surrounding marrow looking as a sheath in B, leaking marrow that is leaking myelin in C. Therefore, Hanover demonstrated that nerve fibers were cell parts. Studying cell parts and their relations led to the neuron theory, but a previous step was the discovery of nerve cell axons by Dates. Otto Dates was an anatomist from Bonn working with Max Schulz. In 1860, he defined axis cylinder, that is, an axon, and he distinguished it from other protoplasmic processes emerging from the nerve cell, that is, from the neuronal soma. He postulated that protoplasmic processes fuse and form a continuous network, thereby believing in what would be later called reticularism as a brain theory on its structure and functioning. Dates died in 1863 from typhoid fever at the age of 29, and his work on the nerve cells of the spinal cord and other structures was edited and published by Max Schulz. This figure of a nerve cell by Dates is one of the first descriptions of what will be later seen as a neuron, that is, a nerve cell with dendrites and one axon. Now, when did glial cells were discovered, and after or before the neuron concept emerged? In fact, Schwann, who established the cell theory, also discovered some kinds of glial cells early. But he could not differentiate them from nerve cells when he made his observations. He observed nuclei along peripheral nerves in their sheaths, corresponding to what we still call Schwann cells, but the Schwann cell concept was built in the following decades. Schwann discovered associated nuclei surrounding peripheral nerve fibers, which represented nuclei of the glial Schwann cells surrounding the fibers and responsible for the formation of the myelin sheath. Other glial cell nuclei were later discovered in other preparations. For example, Jacob Henle was a German Jewish pathologist, a student of Johannes Peter Miller. He knew Remarque and Schwann. He made important anatomical discoveries, among which he described kinds of glial cells. As a Jewish, he was not given a chair in a major German city. He was first prosector in anatomy to Johannes Müller in Berlin, and he held the chair of anatomy at Zurich in 1840, in Heidelberg in 1844, and finally in Göttingen from 1852 to his retirement. He was elected as a foreign member of the Royal Swedish Academy of Science in 1870, but he lacked recognition in his own country because of anti-Semitism. And they described sheaths, that is, you see them in the bottom images, made up of connective tissue with nuclei in myelinated fibers in the front sciatic nerve. He could distinguish the inner sheath, the outer border of the sheath, and their nuclei. Now you see on the top images what Henle also described in ganglionic cells covered with nuclei from satellite cells, which represent types of glial cells. It is interesting to note that glial cells were originally discovered as nuclei surrounding nerve fibers or nerve cells only, since they could not be stained at that time. Therefore, their cytoplasm thus remained invisible. Only progressively did these nuclei became to be considered as signs of glial cells. The nuclei of glial cells were noticed as peculiar anatomical details without much discussion about their role. The discussion on the function of glial cells originated in the work of a pathologist who wasn't aware he was observing a new type of cell in the nervous system. He thought he was observing a class of cells from the connective tissue of the nervous system and in the nervous system. Rokitansky was a Czech pathologist working in Vienna. He studied the morphological changes of diseased cells in various pathological conditions in the nervous system, as hydrocephalus or cerebral hemorrhage. In hydrocephalic subjects, he looked at the brain ventricles and at what was thought as its skin or epithelium, that is the ependyma. Rokitansky saw 5 tissue layers in the ependyma. he considered ependyma was altered in some diseased subjects in a manner similar to an inflammation process as generally seen in connective tissues. Rokitansky convinced himself that the ependyma was thus not an epithelium but a kind of connective tissue, that is, a scaffold or a kind of glue between nerve cells, since nerve cells occurred inside the ependyma tissue. A similar reasoning was made by the famous Berlin anatomist Virchow. Virchow studied in Müller's laboratory. He became the German leader in pathological anatomy in Berlin. He is known to have established the second law of cell theory, according to which any cell comes from a previous cell. But this law was derived from the work of Jewish Polish-German anatomist Remarque, generally not recognized for this. In 1846, Virchow studied the ependyma. He first pointed to the opinion of Valentin and Purkinje, according to which an epithelium covers the nerve substance in the spinal cord. He then followed the original ideas of Rokitansky without mentioning his name. Since the ependyma could react as a connective tissue in pathological conditions, as observed by Rokitansky, it could be seen as mastic or cement or glue between nerve cells, as did Rokitansky, since ependyma seemed to be a connective tissue surrounding nerve cells. That was the reasoning also of Virchow. Virchow therefore concluded, this particular property of the ependyma led me to find a new name for it, neuroglia. The neuroglia concept was thus born in the study of the ependyma, considered as an epithelium and then a connective tissue of the nervous system. Virchow described glial cells, in fact cells from the connective tissue of ependyma, but it is unclear whether his drawings show real glial cells or not. Other types of glial cells were discovered, but independently of the new neuroglia concept at first. German anatomist Heinrich Müller studied with Hanley and Rokitansky. He became professor of comparative anatomy. He studied the eye and described particular cells in the retina and cell fibers, later called Müller fibers, which are glial cells processes or radial glia. However, all these observations of glial nuclei and glial fibers were not yet related to Virchow's concept described in the ependyma. The story of glial cells is highly complex. Camille Golgi described cells resembling spiders between nerve cells. He considered them as a kind of glial cells. He called them multipolar glial cells. Golgi used a new method to stain these cells in 1875, but he had studied these cells before with hematoxylin and carmine in 1870 and 1871. Golgi could describe perivascular feet as glial cell processes. He thought these processes transfer nutrients to nerve cells from the blood. He also used his other mercury chloride method in 1879, and he was able to distinguish other different types of glial cells. At that time, Germany was a world leader in organic chemistry, where new dyes were developed for industry. Biologists took advantage of these new dyes to stain biological tissues. Carl Weigert, professor of pathological anatomy in Frankfurt in 1884, pioneered the use of hematoxylin to stain the nervous tissue. He also stained tissue with the new bacteria stain aniline dyes. Weigert wrote much on the staining of bacteria in microscopy. In 1884, he introduced a precise method for staining myelin sheaths. His new method stained nerve fibers, leading to the discovery of new nerve fiber tracts. Weigert also introduced a staining selective for astroglia, fluorochrome methyl violet, but it stained strongly glial fibers only. Thus, Weigert described a kind of fibrous glia, later called astrocytes. You can see here that the anatomical elements described at that time as these fibrous glia were often in part artifactual, at least in their description, since they were built according to the chemical properties of dyes and not according to the properties of the natural elements, that is, classes of glial cells we know today. The coloration of fibrous glia was a property of the dye to stain the fibers only. Note that this artifactual aspect of cell classifications remain in the discovery of new classes of cells today with new methods. This remark also concerned the Spanish anatomist Ramon y Cajal. He found glial cells with processes in the spinal cord with a new staining procedure, as you can see in these images. And he also found a third kind of glia he considered made up of connective tissue only. Due to his staining procedure, he could only stain the cell body of this last kind of glial cells, which, for this reason, he thought they lacked protoplasmic endings. He called this kind of glia the third element of the brain substance. His student, Del Rio Ortega, was fired from Cajal's institute because he found a dye showing that Cajal's third element was made up of glial cells with processes. Cajal failed to reproduce the technique of his students and his results, and was therefore convinced he was totally wrong. In fact, Del Rio Ortega had discovered the equivalent of the Schwann cells in the brain, that is, oligodendrocytes. He also found that these glial cells could be dissociated into two classes of cells, oligodendrocytes and microglia. In parallel to the slow process of discovery of glial cell entities, anatomists were concerned with their possible functions. For Rokitansky, we saw glial cells were a scaffold and a glue for nerve cells. For Virchow, glial cells also were a kind of glue. To the opinion of Schleich, it is interesting. For Virchow, glial cells also were a kind of glue. The opinion of Schleich is interesting, although it was not much publicized when it was published. Carl Ludwig Schleich was a German physician concerned with painless surgery. He was convinced brain mechanisms were either excitatory or inhibitory, in agreement with 19th century brain theory on inhibition and in agreement with the work of Ivan Sechenov in particular. Schleich believed inhibition could not be a nervous process, since nerve cells were seen as transmitters of excitation only. Therefore, inhibition was seen as a sort of disruption of nerve cell function. Cajal, for example, thought inhibition was perhaps a kind of interruption of nervous transmission by mechanical forces. For Schleich, glial cells were seen as responsible for the inhibition of nerve cells. His original theory is both intriguing and interesting from an historical point of view in the light of what is known today on the role of glial cells in nervous transmission. Current theory were different at the time of Schleich and Cajal. Cajal thought, for example, that glial cells could control nervous activity during sleep, and XIXth, for example, felt that glial cells had no function on nervous transmissions since they were considered as passive electrical elements. In the 20th century, the Canadian neurosurgeon Wilder Penfield reproduced the histological observations of Del Rio Ortega in his laboratory in 1934. He became convinced that oligodendrocytes, as Schwann cells, were responsible for the myelination process of nerve fibers in the brain. This was in agreement with the function of Schwann cells in myelin formation of the French anatomist Louis Ranvier at the end of the 19th century. Also, Penfield was convinced that microglia was made up of wandering scavenger cells. After World War II and the rise of modern biology and early neuroscience, electron microscopy showed that the extracellular space between neurons was entirely filled with glial cells, thus seen as responsible for homeostasis of the extracellular space. But this was due largely to an artifact, the glial cell swelling masking the extracellular space. Also, electrophysiologist Steve Kuffler established a theory where glial cells were responsible for potassium buffer homeostasis, thereby controlling neuronal excitability. Kuffler considered glial cells also important as a scaffold in myelin formation, in neuronal glial cell signaling, in the regeneration of nerve fibers, and this was in agreement with the studies of Ranvier. Kuffler was therefore here synthesizing the knowledge of the known function of glial cells of his time and adding a biochemical trend related to the electrophysiology of neurons. Intracellular recordings had shown at this period that the resting potential of neurons was correlated with extracellular potassium concentration, as was their excitability. Today, molecular analysis show new roles of glial cells in maintaining the functional integrity of axons, in preserving the hematoencephalic barrier, and many other phenomena, including cellular metabolism and glial cell neuron interaction, metabolic interaction, pathological processes, and neuronal plasticity. As you see from such studies, glial cells and neurons are so closely interconnected in different ways that probably new functions of glial cells will be discovered and differentiated among different classes of glial cells in the brain.